now that we've talked about sample prep for the Z1, let's talk about the Z1 light sheet itself. And what is the Z1 light sheet really ideal for? And the Z1 is ideal for, like we talked about, cleared samples. It's also great for organ or whole organ embryoid imaging. As you can see here, we're looking at a whole zebrafish captured by the Wimby Lab and Brittany. It also can image large fields of view, as demonstrated here. And we can also do live samples. We have people who do live zebrafish. We have people who have done live organoid cultures. The system is really, really flexible there. And that's where the Z1's really great at. So let's talk about some quick specs on the Z1, right? Because I know you guys all have these questions about your specific project. So the max res we can do on the Z1 is about 250 nanometers with the 20X, or about 3 microns with the 5X. And just so you have a frame of reference, this is an E9 embryo captured by Lynn from the Magnuson lab. And this is a 5X imaging objective. The max resolution in Z for the 20 is about 650 nanometers. The system is fairly fast. It's not any particularly faster than like a spinning disk, but it is fairly fast. It's not terribly slow at all. The sheet thickness, so this is how we can define our optical sectioning, essentially. That is tunable. We can tune that to however thin or thick we want in the sample, and we can also tune where we want the sheet to be thick. And then our field of view generally ranges from about 125 microns to up to about 3 to 3.5 millimeters we're able to image, actually. So the Z1 is awesome. And now that we've kind of talked about that, I want to talk about the next system that we have, and this is the SOPI. And this is kind of my baby because I learned, my first light sheet was the SOPI that I learned. So I really have a soft spot for this scope in my heart. And the SOPI is a little different than the Z1. And the SOPI is a little different from the Z1 because it both brings the light through the same objective that it will actually collect the emission light from. So it's just a single objective that we're going to be using. And the way that we actually get around this is that we bring the light in from the side of the objective at a 45-degree angle here, and then we sweep the beam through the sample. So the sample, if the sample's here, our beam kind of sweeps right through the sample. And the other thing that makes this system really, really great is that it's incredibly flexible for samples, right? So here you can see that we have a Toka HIT incubator on here, which kind of maintains everything at our 37 or whatever temperature we desire with some CO2, but we can easily remove that. And it's just a kind of a top-down objective where we could dip into it, any sample that we'd like, really. So what is the SOPI really great for? And here we're looking at a cancer cell migrating through about 12 hours with some data that Andres Silster and Ariana collected when we initially started the system. And the SOPI is great for kind of small model organism systems. It's great for kind of larger fields of view, and it's great for like intravital or ex vivo tissue imaging. So this is a great place where you can take out, you know, a section of tissue or a chunk of tissue, and we can image it for an extremely long time. And that extremely long time that we can image it is due primarily because we have really great reduced phototoxicity. The system is fairly fast in that it has an ideal field of view of about 300 microns by 300 microns. And I say that that's ideal because a lot of the specs that you're going to hear here today are if we had the best sample, if it was the most clear sample, if it was just ideal and infinitely bright and things like that. But, you know, in practicality, though, we are getting pretty close to that 300 micron way with the samples that we do have so far. So let's talk about some quick specs with the SOPI, and these specs will likely change this fall. We are upgrading the system to have a resolution bump. The Biomig group with Brian and John Kozeb, run by Anita, they have wonderfully designed a new way to image with this system. So we're hoping to kind of reduce greatly into below the 1 micron level of XY resolution. So we should have that upgrade coming very, very soon. The resolution for the system is about 100 frames per second. The imaging depth is technically 300 microns, but in practicality, we're only going to get to about 100 to 150 at its max, unless we're looking at a cleared tissue sample on here. And our field of view, again, is that 300 micron field of view. So what is the SOPI really, really good at? And the SOPI is really, really good at this ex vivo tissue imaging, and particularly long duration imaging. In fact, this is the longest I've ever been able to image something without a lot of photo bleaching. And this actually went from 92 hours or a solid 4 days here of imaging. And this is data from Aaron Pelosa, and they're looking at actually, you're looking at is a lung slice with individually labeled cells. And then this is kind of the structure of the lung. And they were interested to see is what's going on with these green cells. But I really, again, I highlight that what we were able to do with this system was image for 92 hours. And that's an incredible amount of time to be able to live image a cell for. So we were really pleased with that data. It was really fascinating and really cool to see. So now that we've kind of talked about the SOPI, I'd like to briefly start talking about the Lattice light sheet. And the Lattice light sheet is probably the one you guys have all heard of. So Lattice light sheet involves using a Bessel beam. So what they do is a typical Gaussian beam, which is what most of our scopes are using, is a little thicker and a little wider. So one way to get that down is to make that sheet thinner, is to use a Bessel beam. So that Bessel beam has a much thinner sheet. But as you'll notice here, there's lots of light that come on the outside of this Bessel beam from these ripple effects here. And that autofocus light will really expose our sample. So what Eric Betzig did was he had an idea that maybe we could take this Bessel beam and put it through a pattern. And that pattern would create an interference between those outside sheets of light that we don't really want, and then just give us one fine sheet of light. And that's basically what a lattice light sheet is. So the lattice light sheet works by having a special optics objective, which we shine our light through. And then I'll draw your attention over to here. And this is a really tight interface between a special optics objective and a Nikon objective that comes in. And what they do is we image right in that sample plane. It's got a practical, so we have a practical space limitation with the lattice, but that's easily overcome. We can image samples like zebrafish, organoids, and things like that on there, but we're not going to be able to get your tissue on a lattice. And that's because to get into this very tight pocket between our excitation and our emissions objective, we have to use this sample holder here. And I'll draw your attention to this little gap right here. And I've got a 35 millimeter Mattech dish and a 50 mil conical tube to give you guys an idea of space. Because when I say 5 millimeters, sometimes people, it just doesn't quite ring with them. So this is the tiny space that we would be imaging on a lattice here. And what we do is we give these 5 millimeter cover slips that we've plasma cleaned, and we give them to you so that you can kind of grow and seed your cells on them. And then we load them into the system. And then when they're imaging, these optics are actually dipping into a pool of your media that is warmed and then now CO2 controlled, thanks to an upgrade from Brian. So let's talk about some quick specs on the lattice, right? So the lattice has a max resolution in the dithered mode, and that's where we move the sheet across. This isn't lattice sim, of about 230 nanometers. It has about a 370 nanometer resolution in Z. We can get a cell volume, roughly. I won't talk about frames per second because I know that that doesn't equate to a lot of people. But given the right sample, our maximum speed for a cell volume is about every half a second or every 500 milliseconds. And that's a full volume through the cell at 230-ish nanometers res. We image practically about 20 to 100 microns deep. The field of view can range. We can set that field of view depending on how fast we actually need to go. Is typically what we do is about between 10 to 30 microns. We can get up to 30 or 60 microns as well. We can make that field of view slightly larger, but that's where we go. And again, we mount on that 5 millimeter cover slip that I was talking about. And when we mount the cover slip, we don't actually get all of that 5 millimeter real estate. We're really just left with like a 3x2 kind of circle in the middle of the cover slip. Excuse me. So once you have your data on the lattice, a big part of it is processing. And what I want to draw your attention to is this beautiful data collected by Ali in its MitoTracker Red, looking at mitochondria in the lattice on epithelial cells. And it was really awesome to see. This is a great sample for the lattice. So let's talk about data processing class. When you get your data, it'll come off as several TIFFs per channel per time, or every Z stack per channel per time. And what that basically is just our raw data, but we can't really image that data without really processing it. So we use a program developed by Tally Lambert up in Harvard, and it's called LLS Pi. And this is something that I will do for you guys when you bring your samples to the Lattice. So you'll collect your data. I will process this data for you. And what you'll end up getting is a deskewed folder. So that's where we kind of, the image, because we capture the image, we illuminate at about a 35 to 40 degree angle. And then we capture directly 90 degrees to that. We do have to do like an affine transformation to get that image to look as it does it correctly in nature. And then that's called a deskew. And then what we do is we run it through a deconvolution program, and we're able to adjust all of the parameters of this deconvolution program, as you might expect. And then what you're left with is a decon folder of your TIFFs that we can talk about and work through. So what is the Lattice really, really good for? And the Lattice is amazingly fast. It's probably the fastest microscope. It is the fastest microscope I can think of that we have on campus right now. It has incredibly reduced phototoxicity to it. And I want to draw your attention to both Matt's data over here, which is looking at Afactin, or M-Cherry Espin cells growing on some cell cluster here. And then also Christian de Cassiger's data from the Mercar lab. And they were interested in looking at how endosomes move around in a cell. So previously, before, we were actually getting a lot of people that are interested in this particular topic because the Lattice is so fast. We can image at volume, and we're actually able to track some of these functions that occur. And they're particularly interested in how these little tiny dots, which are whipping around our cells here, are moving out to the apical membrane. This image here is captured just shy of our maximum speed of about a half of 500 milliseconds per cell volume. And this data is in 3D.